Hello guys, welcome to this course. So this is the last lesson of this course, and in this lesson we'll be studying about Golgi apparatus. This course is presented by me, Navneet Sharma. You can know more about me from here. You can follow me on this link, and if you like the lesson, kindly rate, review and recommend. The Golgi complex. In the animal and plant cells, layers of fat-filled structures are present. These are called the Golgi apparatus or Golgi complex, okay, or Golgi bodies. In 1898, Camillo Golgi recognized it in the nerve cell of the owl. The Golgi complex of invertebrates is called as the dictyosome. Apocrine's Golgi complex is seen in all eukaryotic cells. It is not seen in mature sperm, RBCs, and prokaryotes. Okay. In prokaryotes, you know that these cell organelles are absent, so Golgi apparatus is also absent. This is a diagram of Golgi apparatus. Okay, so we have a cis phase and a trans phase. So we have some cristae, and some secretory vesicles. Okay. The Golgi complex occurs in two forms. First is localized form. Second one is a diffused form. The localized form the Golgi complex occurs singly and has a fixed position in between nucleus and secretory board. In the diffused form, in the nerve cell and liver cells, Golgi complex is scattered, but in each unit is called as dictyosome. Structure. Golgi body is seen in the form of three components. First is cisternae. These are tubular, flat, fluid-filled sacs. They show 200 to 300 angstrom width. Each sac is covered by two membranes. In a dictyosome, 3 to 7 cisternae are present. They are arranged one above other. Their convex side is towards nucleus and their concave side is towards the plasma membrane. Okay. Next we have the convex side of cisternae is called as the forming phase. The concave surface is called as the maturing phase. It shows big secreting vesicles. These secretory vesicles store secretory substances. They may develop into the lysosomes. Okay, I've already told you in the previous lesson that lysosomes are formed from the Golgi complex. Next we have the polarity of cisternae. The cisternae shows maturing phase and forming phase. The forming phase is convex and towards nucleus. The smooth ER gives vesicle. They unite to form the cisternae. Next we have Golgi vesicles. On the forming phase of Golgi cisternae are small vesicles are present. They are 400 angstrom in width. They usually develop from endoplasmic reticulum. Okay. Next we have the secretory vesicles. On the maturing phase of Golgi cisternae, secretory vesicles are present. They contain secretory products of Golgi. They finally change into the lysosomes. Okay. Now we'll move towards the chemical composition of Golgi complex. First of all, it contains phospholipids. These substances have a composition which is in between the structure of phospholipids of endoplasmic reticulum and the plasma membrane. Enzymes, ATPase, CPTase, transferases, etc. enzymes are present. Carbohydrates are also present, in which glucose, mannose, galactose, etc. carbohydrates are seen. Then we have the origin of Golgi complex. First of all, from ER, AIIMS and Castle in 1968 proposed that Golgi system cisternae arise from endoplasmic reticulum. Okay, there are various theories, so this is one of the theories proposed firstly. Secondly, rough ER produces protein which will be transferred to the smooth ER. The vesicles are developed from the ER. By the fusion of these vesicles, cisternae are formed. From the cisternae, secretory vesicles are maturing phase. These sites are called as GERL, that is the Golgi endoplasmic reticulum lysosome, okay, which will form the lysosome on the later stages. From nuclear membrane, there is a second theory which was given in 1965 by Bausch. He described the origin of Golgi from the outer membrane of nucleus. Vesicles are pinched off from the outer nuclear membrane and they are united to form the cisternae. Okay. Thirdly, we have by the division of pre-existing dictyosome. During the cell division, the number of dictyosome will increase. In each daughter cell, the number of dictyosome will be equal to the dictyosome of parent cell. Okay. Next, we have zones of exclusion. Each Golgi complex is surrounded by a specific zone of cytoplasm. In this zone, ribosomes and other organelles are absent. Such zone is called as the exclusive zone. Okay, the ER present in this zone will show smooth surface. Now the functions. Golgi complex is mainly concerned with the secretory function. In different types of cells, different types of secretions are produced by the Golgi complex. Now the Golgi complex and secretion. The secretory mechanism will follow these processes. Firstly, proteins are produced by the ribosomes. Then they will be transmitted to the smooth endoplasmic reticulum. From there, they are concentrated at Golgi complex. Now from the Golgi complex, the secretory vesicles are formed. They produce the secretion. These secretion are of three types. First is holocrine secretion, and second is apocrine secretion, and the third one is merocrine secretion. Okay. Now the holocrine secretion, the entire cell is filled with the secretory product. It gives rise to the secretions. Example, sebaceous gland. The next is apocrine secretion. The secretory vesicles will come out of the plasma membrane. They take away the small amount of cytoplasm. Example is the apocrine sweat gland. Next we have the merocrine secretion. The secretory vesicle will unite with plasma membrane and open to the exterior. Example is the salivary gland. Again, these are the cell type and Golgi functions in different type of cells. In the pancreatic cells, they function as the digestive enzymes. In hepatic cells of liver, they help in the secretion of lipids. In the plasma cells of blood, they help in the formation of immunoglobulins. In the thyroid gland, they help to produce the thyroxine hormone. Now the synthesis of glycoprotein. Most of the cells will produce glycoproteins. The glycoproteins contain a protein part and a carbohydrate part. They are the important for secretions. Now the storage of secretory products. Ribosomes synthesize proteins. They are discharged into endoplasmic reticulum. They are concentrated in the cisternae. Then they are stored in the secretory vesicles. Thus the glycoproteins are stored in Golgi body. Okay. So the formation of a lysosome. From the maturing phase of cisternae, granules are produced. They are united to form the lysosomes. Okay. Acrosome formation. During spermatogenesis, Golgi complex will develop into small proacrosomal granule. Inside each vacuole, it increases in size and they unite to form a single granule, single proacrosomal granule. This acrosomal granule is formed and it forms the acrosome. This acrosome helps the sperm to penetrate into the ovum during fertilization through the help of some enzymes. Okay, so this course ends here. So I'll be making some new courses as well. So I keep learning and thank you guys for watching this lesson.